Hi, everybody. Thanks very much for listening in today. I'll be talking through the latest activities at Arovella Therapeutics. And just a quick reminder, we're working on our invariant natural killer T cell platform and utilizing that to tackle various forms of cancers. I will be making forward-looking statements, so please do take note of our disclosure. Now, just to jump into the highlights, so we are working, as I said, on an invariant natural killer T cell platform, or iNKT cell platform for short. And we're utilizing that to tackle both blood cancers initially, but looking forward to expanding into solid tumors. Our lead product is by the name of ALA-101, is our treatment for what we call CD19 expressing blood cancers. So that means that on the surface of those tumors, they have CD19 on their surface, which we can target. And we're expecting to take that into phase 1 clinical trials in 2024. Now, the cell therapy sector has certainly revolutionized the way we think about cancer treatment, but we do know there are some challenges that still hamper the sector, and we do believe our iNKT cell platform will play a role in alleviating some of those challenges. Now, in addition to our technologies, we've amassed or assembled a very strong leadership group with a lot of experience broadly across drug development, but specifically also around cell therapies, which is very important for this type of technology. Now, in terms of the company and where we've positioned ourselves, we've relied a lot on the use of strategic acquisitions to bring in what we think are cutting-edge technologies, and we'll continue to do that as we move forward, not in a scattergun approach where we're looking to get quite different technologies, but really where we see that there's strong alignment with what we're trying to do with our iNKT cell therapy platform. Now, in terms of an iNKT cell platform, we are the only company in Australia working on this type of cell technology and one of a handful of companies globally. So we do see that we have a fairly unique value proposition moving forward. Now, in terms of our financials, we are currently capped at around the $60 to $65 million mark. This is a little bit old, this slide, and we currently have about $7.4 million cash pro forma after completing a share purchase plan at the beginning of July. So we're not looking to raise money. And in terms of our shareholders, we're delighted that we've got Merchant as our largest shareholder. I'm pleased with the level of support that we've received from a range of high net worths and also smaller institutions, but of course, always looking to welcome new investors to the register that understand the risks that come with a biotechnology investment, but of course also the potential rewards that can be achieved. In terms of our share price performance this year, it has been quite strong. We were happy with the level of shareholder appreciation and support that we saw in April after we presented some data at the American Association for Cancer Research in Orlando. And of course, we're continuing throughout the remainder of this year. We've got quite a number of data points to come out, and we're so excited to announce some of those before the end of the year. And of course, looking forward to creating additional shareholder value over that time period. Now, in terms of the sector, I think more broadly biotech, we have seen it's been a pretty challenging 2-year period with a lot of companies, obviously share prices being suppressed quite heavily, and a number of companies actually in the US particularly trading below their cash value. But what we have continued to see, particularly in the cell therapy space, is there have been a number of either acquisitions or strategic licenses that have continued to take place, with already 4 this year, which is quite promising given the state of the sector. And what's important to note here is that a lot of these transactions are not just that late stage phase 2, phase 3. In fact, the ones that I've demonstrated here are really quite early, either being preclinical, so that's before they've actually gone into humans, or phase 1, with one there itemized here as phase 2. So we are seeing quite a lot of activity at the early stage, and we think that comes off the back of the fact that for all 6 approved cell therapies to date, they have actually been approved after phase 2, and those have become the registrational or pivotal trials for those products. So we think that's actually bringing the likelihood of commercial transactions a little bit earlier, because we're able to capture data in trials like phase 1, where we don't go into healthy volunteers, we go into patients directly. So we're able to capture some data or some readouts, let's say, those earlier clinical trial stages. And just important to note that some of these deals have been quite sizable, with a number of upfronts in the sort of $100 to $200 million range, and some interesting transactions more recently. And it was exciting to see Imugene in-license a cell therapy from a US company with a total deal value of around the $227 million mark. And this one's particularly interesting, Arovella Therapeutics and Astellas, where it was actually a $25 million upfront payment and also a $25 million direct equity investment into Poseida. And that was for an exclusive option for negotiation. And one last example was Janssen and Cellular Biomedicine Group in August, where that was a $245 million upfront payment again to get access to their phase 1B technology. So good that we're still seeing a lot of activity in the sector. We think that's important moving forward, and we're certainly looking forward to seeing how that continues throughout 2023. Now, in terms of where we sit in the cell therapy sector, it's important to point out there are a number of immune cell types that people are working on. So T cells and NK cells are the most well documented. And you can see here, just by virtue of some of the companies that are working in the space, and this is a snapshot, that it is quite a crowded area with hundreds of companies and likely thousands of clinical trials ongoing. So contrast that with the iNK T cell space, and as I mentioned earlier, it is a lot smaller. There is only a handful of players globally, and it's great that Arovella is one of those players and continues to be kicking goals as we move and look to take our products into clinic in 2024. Now, just to step back a little bit, so what is it exactly that we're working on? And I'll start by explaining the original generation or original version of the technology, which is CAR-T. And as I just mentioned a moment ago, they rely on the use of a specific immune cell type called a T cell. And what actually happens, and just to be clear, so T cells they're almost like the soldiers in our bloodstream. If something that enters that doesn't belong, whether it be a bacteria, a virus, or in some cases cancer cells, it's the job of the immune cells, in this case the T cell, to eliminate those foreign objects. And so what we actually do for this type of therapy is the blood is taken from the patient. So it's the patient specifically that would have presented with this particular disease. Their T cells are isolated from their blood. They actually get shipped off into a laboratory where they can undergo a genetic engineering step where we introduce what's called chimeric antigen receptor, or CAR, and that's where we get CAR in CAR-T. And if you think about those as heat-seeking missiles or guided missiles on the surface of that immune cell that are able to now recognize and terminate certain types of cancer cells. So then once that's done successfully, the cells get grown up. So we've got millions of them. They then get given back to the same patient that actually the raw material was essentially collected from. And when we see a particular cancer cell, in here we've got green bumps, which would recognize would represent the target. We have the immune cell finding that target, and then, as I said, that will cause the elimination of those cancer cells. Now, it has revolutionized the way we think about blood cancer treatment because, as I said, we've seen pretty remarkable results to date with both cases in pediatric settings here. This is a young lady, Emily Whitehead, who now it's 11 years more recently that she celebrated as being cancer-free, but also in the adult setting, we've seen similar results as well. Not surprisingly, because of that level of activity, the cell therapy market, the products available have already generated strong sales, and it is expected to be a multi-billion-dollar sector by 2030. Now, one of the downsides to CAR-T therapy is that, unfortunately, while some people may display great results and continue to live on cancer-free for quite some time, we still do see that there's about 40 to 60% of patients actually relapse post-CAR-T therapy. Now, in addition to that, we also know that there is some pretty major supply challenges for this type of therapeutic. Now, if you remember back, I just explained that the T cells must actually come from the patient that is to be treated with that particular product. And what that means is that every single dose of the therapeutic has to be made patient-specific. So that's very inefficient logistically, and what it does is it drives up the manufacturing and supply chain costs. So on average, to get access to this type of therapy, it's about $500,000 per dose per patient, and that's US dollars. Now, in addition to that, because the people that are actually going to take the therapy have actually seen the disease, they have been exposed to things like chemotherapy, it stands to reason that the starting material, their T cells, is actually compromised. So you actually have a reduced efficacy due to that compromised starting material. In addition, not all treatment centers are actually able to collect the cells and manufacture the product. So this does mean that there's a number of patients that may be actually eligible to take the therapy, but they won't actually be able to get access to it. In addition to that, it takes about 4 to 6 weeks for each dose to be made for the patients. So what can happen is patients with particularly aggressive forms of the disease can actually die while they're waiting for the treatment. And of course, because you're making the therapeutic for every single person every time, it really opens up this risk of manufacturing run failure, which further increases the time to treatment, which can increase costs and of course the likelihood that patients won't survive to take that therapeutic. So as I mentioned, we do believe that the CAR-iNKT cell platform that we're developing has the potential to address some of these challenges, but also from the data we've shown with the potential for improved efficacy. So I'd like to introduce invariant natural killer T cells very briefly. Now, as I mentioned, T cells are the mainstay of cell therapy to date, CAR T. Others have tried to work on natural killer cells as well with limited or varying degrees of success. But for us, it really is the iNKT cells that we're most interested in for a number of reasons. Firstly, if you look here, there's actually 2 circles delineating both what we call the innate immunity, which is our fast-acting immune system, and then there's a second circle delineating the adaptive immunity, which is a longer-term, more durable response. And what we see is that iNKT cells actually bridge both the innate and the adaptive immune responses, so they're capable of assembling a fast or a more prolonged response. They actually contain the ability to kill like both T cells and NK cells, and they actually naturally target and kill certain cancers that produce CD1D on their surface, and they do that through what we call the invariant T cell receptor, or ITCR for short, which is shown here in red. And more importantly for us, because of this ITCR, and I won't go into the detail here, but what it means in contrast to a classic T cell is iNKT cells do not cause graft versus host disease, which is when an immune cell, if it was given from someone to another person, those immune cells would actually start to attack other parts of that particular person that received the therapeutic. iNKT cells don't do that, and that's what enables us to be able to give them from a healthy donor to many patients. So again, we think that's a really big advantage and will go a long way to alleviating some of the issues with the supply chain. Now, in addition to that, iNKT cells have additional anti-cancer properties. They shape what we call the tumor microenvironment. They also infiltrate tumors, and they can activate other components of the immune system to join in and kill cancer cells. So just showing that on a diagrammatic level here. So as I mentioned, we've got 2 ways of iNKT cells to kill particular forms of cancer, those that produce CD1D. We have the ITCR, which will recognize that and trigger their destruction. And we also have the chimeric antigen receptor, or CAR, which we introduce, which will find the target on the cancer cells. And when that happens for either, we get the release of components which triggers the cancer cells to be killed. And we also know that iNKT cells can block or hamper tumor-associated macrophages or myeloid-derived suppressor cells. And these are pro-cancer, so they actually support the cancer cells to grow. So again, iNKT cells have the ability to regulate those players. And as I said, iNKT cells also, through the release of signaling molecules, can activate components of the host immune cells like T cells and NK cells. Again, all contributing to having multiple ways to combat the cancer cells. So when we think about manufacturing, this is a really important part of the therapeutic strategy. So in contrast to the workflow I showed previously, instead of starting from a patient, we will actually start from a healthy donor. Instead of isolating the T cells, we isolate the iNKT cells. They get shipped to a lab still, where we reprogram them to express the chimeric antigen receptor. We grow them up into billions, and then they all get vialed and stored in a freezer so that they're manufactured and waiting ready for use. And then when a patient is eligible to take that therapy, we will literally thaw those cells out, and then they'll be able to be given to the patients that are eligible to take the therapeutic. So we believe there are a number of advantages. One, healthier starting material, because we're starting with a fully functional immune system of a healthy donor. It's scalable in terms of manufacturing because we can get multiple doses per batch. Because we're not manufacturing in real time, it means we have faster access to treatment, so patients can take that therapy immediately. And because it's sitting in the freezer ready for use, it essentially eliminates the risk of manufacturing run failure. So I'd like to just talk more about our lead product quickly, ALA-101, which is a next generation off-the-shelf cell therapy for CD19 expressing cancers. And just to be clear here, this is still quite a significant population. For things like non-Hodgkin's lymphoma, it's in the order of 60 to 80,000 patients diagnosed with this type of cancer type in the US alone each year. And what we've seen is there are a number of treatments that are available for these patients. But in terms of the CAR T products, it's great that they've demonstrated promising activity and they have started to move up into second-line therapy. But what we still see is there are actually a number of patients that do relapse from that therapy. Of course, there's some side effects associated with this type of technology, and of course, the supply chain issue of having to take the therapy from the patient or start to manufacture the product from the patient. So we do believe that ALA-101 has a really important role to play because we're able to use it off the shelf. We can start with healthy donor material and get the therapy to multiple people. And of course, we do think that it's got an excellent role to play, and looking forward to starting phase 1 clinical trials in 2024. And in terms of why we're so excited by the platform, I'd like to just quickly run through some of the data that we've generated to date, where we're looking at a particular cancer type here, or a tumor cell that produces both CD19 and CD1D, so both the targets of iNKT cells. And these are administered into the mice. And so we're looking at day 0 here. So this is after the cancer cells have been engrafted. And then we're looking at the bottom 3 days after treatment with a range of treatments. Anywhere you can see the color, the red, the blue, the yellow, or orange, that's actually cancer cells inside of the animals. And in the first column, we're looking at phosphate buffered saline, or PBS, which is essentially salt water. Then we're looking at T cells, which lack the chimeric antigen receptor. Same for the iNKT cells, so it's cells with no targeting. Then we're looking at CAR T cells with a chimeric antigen receptor targeting CD19. So again, this is more closely related to what's currently approved and on market. And then in the last column, we're looking at ALA-101. And I think what's quite clear is that we see that the cancer cells in the ALA-101 sample have actually largely been eradicated or eliminated, in contrast to the control groups, where you can still see there's quite a lot of color remaining, demonstrating there's a lot of cancer cells still persisting. So this is in line with what we expected and what I showed earlier, that iNKT cells mount a very fast, very quick and rapid response against foreign objects like cancer cells. So the most important part of this, though, is that we actually take this a step further and see, great, we get fast elimination of the cancer cells, but does that actually translate into longer term survival? So what we're looking at here, it's called a survival curve. On the vertical axis, we have how many animals are alive, with 100% alive, the line would remain at the top. And of course, with 0%, that would mean all the animals have died. And then we're looking on the horizontal axis. Now, instead of 3 days, we're going out to 90 days. So in the black, blue, green lines, we can see that all the animals have actually died between 40 to 60 days. In contrast, we see really impressive data here for the CAR T cells, where it's about 60% of the animals are alive at 90 days. And then what we see is just the iNKT cells happen to have better activity or better responses here in terms of animal survival, with about 90 to 95% of the animals alive at 90 days. And taking this a step further, this was an interesting finding that came out of these studies, where 4 of the mice, so 4 out of the 19 mice treated with the iNKT cell therapy, they actually looked like they were cancer-free at the early time points. And what actually happened between 20 to 30 days in all 4 mice is the cancer cells actually returned. And you can see it here. It's quite clear that it's localizing to the brain of these particular animals. So just to be clear, as I said, that was 4 mice treated with the iNKT cell therapy, and that was 4 out of 19. Now, naturally, what would be expected here is that because the cancer cells have returned, that the animals will also go on to die from the disease. Instead, this is not what happened. And again, just to be very clear, there was no further dosing of the iNKT cells. But what we saw was what we term now a spontaneous secondary remission, where the cancer cells were actually eliminated a second time. So we do find this quite interesting because it tells us a range of things. Firstly, we believe that the cells are able to remain long enough in the body so that they're able to eliminate the cancer cells a second time. And if that's the case, then they're actually crossing into the brain, so passing through the blood-brain barrier, and also that they're able to have activity on a more solid tumor-like cancer that has largely entered into the brain. So when we presented our data at the American Association for Cancer Research in Orlando this year, we did repeat much of this work, and we were able to show that our manufacturing process facilitates the production of large numbers of potent CAR-iNKT cells. Again, we think that's a really important point when we think about having multiple doses produced from a single batch, so that we can alleviate some of the issues that we've seen for the costs and the logistical challenges for the current CAR-T therapies. So we've started to make the therapy using lentiviral vector system with a proven safety profile, so it's third generation, and that sets us up nicely for the preparation of the manufacture of clinical grade material. And again, when we've used this process, where we've used those reagents, we've actually been able to show, again, in a really aggressive model of human leukemia, a B-cell acute lymphoblastic leukemia cell line, that we do still see significant extension of lifespan, which confirms, gives us the confidence to progress ALA-101 towards first-in-human clinical trials. Now, as I said, we are very interested in taking that product into clinic, but we're also very interested in leveraging the iNKT cell platform to target solid tumors for a few reasons, but a big one is the fact that 90% of newly diagnosed cancer cases are actually some form of solid tumor. So we see that there's still an enormous societal need and requirement for better therapies against things like solid tumors. And so we're very excited to move into this space. So to do that, as I said, what we're doing, we're still going to work with our iNKT cell platform. Initially, we've been using that to tackle what we call CD19 positive blood cancers, but now we've got a series of strategies that we're actually employing so that we're shining a light on many more different cancer types that all constitute different forms of solid tumors. Now, just to be clear, solid tumors do pose unique challenges to cell therapies that are quite different from a blood cancer. So on the right here, for blood cancer, for example, it is in a liquid setting. So those cancer cells are quite accessible by the cell therapy. So you can see here there's quite a lot of room for the cell therapy to get access. And we do believe that's a part of the reason why we've seen such excellent activity for CAR-T cells to treat different blood cancers. And of course, once you get engagement, we get expansion of the particular cell therapy, which drives the prolonged response. In terms of a solid tumor, it's a little bit different. You see that they form a more mass-like structure, so it's harder to get access to it. We have what's called the tumor microenvironment, which complicates things, and we have antigens that are not as specific and uniformly expressed on the cancer cells. So that makes it quite a challenge to find the right targets to go after. And as I mentioned, the tumor microenvironment also contains pro-tumor cells like myeloid-derived suppressor cells, tumor-associated macrophages, which also support tumor growth. So we do believe that, as I mentioned earlier, iNKT cells have a number of unique properties over other immune cell types that we think contribute to the fact that they should have a good role to play in the treatment of solid tumors. So as I mentioned, we have the killing by the ITCR engaging with CD1D, the CAR engaging with the target we tell it to, but we also have the fact that iNKT cells naturally home to tissues, and they've actually been documented in human clinical trials to infiltrate into tumors. As I mentioned, they block and kill tumor-associated macrophages or myeloid-derived suppressor cells, so they help reduce the level of these cells that actually contribute to that pro-cancer cell ability. And as I said, they also start a cascade of signaling events to help recruit other immune cells that can also start to facilitate killing of the tumor cells as well. So our first strategy to tackle solid tumors is actually using a partnering approach. And our first example of this is working with Immugene's OnCARlytics platform, where they have an oncolytic virus that gets injected into the patient. The oncolytic virus naturally and selectively and specifically infects solid tumor cells. Now, once it's in the solid tumor cells, its job is essentially to replicate and divide and eventually burst those tumor cells. But for those that don't undergo that bursting or lysing, the virus has also been equipped to make those tumor cells produce CD19 on their surface. So what happens then is we administer the CAR-iNKT cells. They then come in over the top. They'll be able to find and recognize the CD19 on the solid tumor cells and then trigger the destruction of those cells. Now, in addition, as I mentioned a moment ago, we have the impact of the iNKT cells on the tumor microenvironment, and also the fact that they can recruit endogenous immune cell components. So we see that there's a lot of or a multifactorial approach here to killing the solid tumor cells. So we did get some promising data earlier in the year in the in vitro setting or in the test tube setting, and we're expecting in vivo data for this program before the end of this calendar year, which is exciting. Now, our second strategy actually relies on the use of bringing in additional or novel CARs that go after different targets that are expressed in the context of a solid tumor. So here, what we're representing is it would actually have a different chimeric antigen receptor that goes in to be expressed on the surface of the iNKT cells, which again gives us a shot on goal for different cancer types. Now, the beauty of this strategy is that we can leverage all of the manufacturing workflow that we've generated to date, so we don't need to repeat a lot of that. The same process would be utilized to generate that therapy, so we do expect that it would be a little bit more swifter getting these products into clinic. One thing that we will need to make for each new CAR that we look to work with is what we call lentiviral vector, and that's how we actually get that particular CAR into the iNKT cells. So again, we think that's a really exciting strategy, and of course, it's something we're pursuing, looking at a range of different targets that we think might be suitable to bring into the company. And the third strategy actually relies on what we call armoring the iNKT cells, which is essentially working to enhance their persistence, or essentially how long they actually last in the body. And the idea being that the more we can increase the persistence, the more that we can increase the anti-tumor activity, because the cells are actually alive in the body for longer. So we do have an exclusive option already with the University of North Carolina for a cytokine technology, and that's been developed by one of the pioneers of CAR-iNKT cell biology, Professor Gianpietro Dotti. And again, similar to the additional CARs, we can incorporate things like this, the cytokine technology, for example, into that the same lentiviral vector. So we don't actually have to require changes to the manufacturing process. We just need that one raw material input that would enable us to utilize a technology of this nature. So we already have collected some in vivo data, so animal data for this particular program, and it confirmed exactly what we thought, where we're actually able to see the iNKT cells expand or grow more and survive for longer than CAR-iNKT cells that actually lack this technology. So looking in the bloodstream, it was actually 10-fold more CAR-iNKT cells at the 4-week mark after treatment in a mouse model, again, which is very compelling. And it supported exactly what we expected after that point, that there would be a superior anti-tumor response. And so what we saw is that 75% or greater than 75% of the mice that were treated with CAR-iNKT cells with that technology were alive at 61 days versus mice treated with CAR-iNKT cells that lacked that technology had all died within 49 days. So again, really impressive data and a really exciting strategy to take forward. And the beauty of it is I think most of these strategies can actually be utilized in parallel, which is nice. So in terms of our pipeline, it is continuing to expand. We have ALA-101, which we're developing for non-Hodgkin's lymphoma or CD19 producing lymphomas or leukemias. We're working with the Immunogene's OnCARlytics platform to tackle a range of solid tumors. As I said, we're working in the background to look for novel targets that can be used to treat a range of different solid tumor types. And of course, the cytokine technology, we believe, has a significant role to play in helping us to tackle a range of different solid tumors, which is, again, very exciting for the platform. So over the course of 2024, fiscal year 2024, we do have quite a lot of work underway. And most exciting, I think, is that we're looking to advance ALA-101 into its first in-human clinical trial during 2024. So that will be a safety study with the primary endpoints being looking for any dose-limiting toxicities. But of course, as I mentioned, because that is a phase 1 in patients, that we would also be looking to see any efficacy signals. But just being clear, that's a study that's primarily designed to look for safety. So for that program, we've got a range of things to do on the manufacturing, so complete the process optimization and scale up in prep for what we call CGMP or clinical grade manufacture. We're expecting in the next quarter to produce the CGMP lentiviral vector, which is a really important milestone for getting the product manufactured. And in the background, we're working to finalize the clinical trial plan for the phase 1 study. So the idea being at the first half next year that we actually complete the manufacture of the product for phase 1 clinical trials. And once that's done, then we complete all the activities required to generate the documentation for things like INDs or investigator brochures that we then submit to regulators to start phase 1. Now, in terms of our other programs, we're excited that for the CAR9 or the ALA-101 plus oncolytics combination study, we'll have data before the end of the year. And we are analysing different CARs that we would like to consider adding to the platform. And for the option to technology, the cytokine technology, we'll look to, pending our due diligence, we'll look to in-license that technology as well. So over the first half of next year, assuming that some of these things do pass our due diligence tests, that we will actually initiate a range of proof of concept studies. And so looking forward to getting some of that data out, of course, as we progress those studies. Now, I mentioned it before, and I won't go through everybody in the team, but it is a phenomenal group of people. I think major point here is that we've got an exceptional amount of experience across drug development, and really it spans all the way from academic researchers through to drug development, but through to corporate transactions through either venture capital or business development or corporate activity in the form of mergers and acquisitions. So it is a phenomenal group with an incredibly strong amount of drug development experience. And as I mentioned earlier, really, really good that there's a lot of experience specifically in the cell therapy sector, which is a pretty unique sector and does require a lot of expertise. So in summary, I'd just like to finish by saying that we have a novel iNKT cell platform that we're using to develop off-the-shelf cell therapies to treat different forms of cancer. Now, as I've illustrated, CAR iNKT cells have multiple anti-cancer properties, both directly but also by influencing things like the tumor microenvironment or the host immune cells. Now, we know that if we're able to produce an off-the-shelf product, there is the improved manufacturing and logistics elements, which we think will significantly drive down costs and make this type of therapy more affordable and more accessible to many more people. So very excited that our lead product, ALA-101, is progressing into clinical trials and expecting that to commence in 2024. But excited that we've now started to embark upon some exciting strategies that we think will help iNKT cells have a prolonged and exciting place in the treatment of solid tumours. And so for all those reasons, we do see that, again, as the only iNKT cell company in Australia and one of a handful globally, that we are really positioning ourselves well for growth over the years to come. And with that, I'd just like to say thank you again, everybody, for listening. Appreciate your time and looking forward to giving you updates on the company as we progress. Thank you.